Okay, I'm here representing our multidisciplinary team from the University of Maryland, and the first slide will probably be very familiar to Dr. Heinonen, and this is really from dramatic work that he and Alison Burgess published in 2011, showing that with safe parameters of blood-brain barrier opening, you could get something as large as neuroprogenitor cells, which are on the order of 10 microns across the blood-brain barrier. And one of the things that was really not emphasized this morning, with all of the emphasis on finding safe and effective parameters for blood-brain barrier opening, is how inefficient this process is. Blood-brain barrier, all forms of those deliveries are poor. Usually 1 to 2% of an injectate gets into brain. In most FUS studies, we're really talking about only sometimes 0.1% or less of an injectate. Even if you put it intracarotid, which is not particularly minimally invasive, will enter brain. So the question is, how can we, within these safe sonication parameters, improve that efficiency of transfer? And another technology that Paul Urowski and I had been working with for a couple of years before that is a technology known as magnetic attraction or magnetic targeting. And with the use of super paramagnetic iron oxide nanoparticles, SPIONs, and they've been used widely for labeling cells in transplantation studies for follow-up with MRI. And most cells in culture will engulf these particles, and the particles we use are particles called MIR-B, and they're not only iron oxide containing, they have a dextran shell, and they're also rhodamine, and we have them both rhodamine and a fluorescein-like label, and cells in culture will engulf them without any effect on their biological activities. The cells will differentiate, they'll divide, and they engulf enough of them that they actually will respond to an external magnet. These particles have several picograms of iron in them. They have up to 30,000 of these particles will be engulfed. And we had previously published that we can actually influence the migration of cells in vivo, and this is again a work, lead author is Dr. Shen, with a so-called magnetic hat, by putting a small, basically refrigerator magnet mounted on the animal skull in the region of a TBI. Without the magnet, in that area that does have a damaged blood-brain barrier, you do have some cells that'll enter, and that's the line in black, and the red line shows this is dramatically greater when both a TBI and a magnet are placed. So like several of the groups, we've been able with our device, our animal device, to create an area of safe blood-brain barrier. It's really more of a column, cylinder that goes through the cortex and into the striatum without significant pathology. And we've been able to deliver, in this case intravenously through a tailbane injection, we've been able to deliver these SPION labeled cells, and we confirmed the identity of them not only with fluorescence, but with iron staining, and since these are human cells, with a human-specific antibody. The SC121 antibody. So we know that the particles have not been phagocytosed by some other host cell. And so we were able to ask the question, if after sonication and injection of cells, if we just allowed the animal to lay on a powerful magnet, could we increase the number of cells? And so we inject a mixture of cells. The green cells are magnetic. The red cells are dummy particles that are fluorescent but without magnets, and we used either a very large, again, neodymium magnet, or a so-called Hallback array, which has less decrement from the source. And what we saw was virtually a dose-response effect. So dorsal means close to the magnet in the cortex. Ventral is in the striatum further away. And with the lower power magnet, a 2x2 inch cylinder, what you have is still an increase in the number of cells if they are in fact iron labeled. And once you increase the power of the magnet, you see a greater percentage, close to 90% of the cells with a more powerful magnet are in fact labeled with the iron particles. So again, you get about a 10-fold augmentation of cells within the brain. And so I think in our conclusion, I think I lost my last slide, but our conclusion is magnetic attraction is a very viable augmentation to increase cell entry with blood-brain barrier disruption. Okay. And again, we're going to be looking at this, and even in a totally non-invasive model, the enhancement of transnasal delivery. Just about.
Any questions? We have time for a couple of questions.
Joe Frank, NIH. Paul, over here. So one question would be, when you looked at markers on the surface of your NSC, did you see any increase in integrins on the surface? Because PFUS plus microbubbles will actually increase cell adhesion molecules on endothelial cells. And that kind of attraction may be something that's important as compared to, say, a dummy particle which doesn't contain iron in it.
We haven't yet. We actually have that tissue, and we plan on doing that probably within the next few days.
But take the cells, just don't even take the tissue. Just look at the cytospins and ask the question, or do a FACS analysis and ask the question whether or not your integrins are going up.
Yeah, that's certainly doable. Thanks.